Thank you, Hassan, for the very warm welcome. It's a pleasure to be here at the Royal Institution. I came here 2 years ago for one of the summits that talked about aging, so it's great to be here talking about my research, which is in the context of aging. So today, I'll be telling you about metabolites and metabolism and how we can learn more about solving Alzheimer's disease. So more than 100 years ago, Dr. Alois Alzheimer first discovered the disease in one of his patients. And what he found out was that this patient's brains had clumps, protein clumps, and plaques that were only peculiar to Alzheimer's patient brains. And it has been 100 years ever since, and we still do not have a cure. So even now, in patients, if you would see a healthy aged brain versus a brain that has Alzheimer's, you can see a distinct shrinkage that you can see on the left-hand side. And there is a buildup of these protein clumps inside and outside the neurons that cause damage to cognitive abilities. But what we do not understand is, are these really the causal factors for the disease, and what really happens? So over many, many years, scientists, industry, pharmaceuticals have looked at various strategies to combat Alzheimer's disease. They've looked at various mechanisms and targeted them to come up with a successful drug. Now, if you have been following the news, you must have seen about the recent approval of aducanumab and lecanemab, the two monoclonal antibodies, that do something for the disease, but there haven't been enough success. So how we see that is almost that you're trying to sweep off the ashes after the forest has already been burned down. But we do not understand what were the causes, why the forest has been burned down, and what led to the ashes. So that prompts us to look beyond the final plaques that we see in these patient brains, and what really happens during aging in people's brains that leads to Alzheimer's disease. To understand that, we go back to the basics, and that's all about proteins. So these clumps that Dr. Alois Alzheimer saw in his patients' brains were formed of proteins that had gone awry, that had just formed these clumps. Now, not all proteins cause disease. Proteins are the workhorses of the cell. They perform most of the functions that are there, ranging from enzymes to building the structural components of this structural components, and also working as signaling molecules. If they were to go wrong, that causes disease. What constitutes these proteins? How do they look like at the molecular level? So these proteins are built with basic building blocks of amino acids in the form of a polypeptide chain. Now, in front of me and in front of you all, there are various structures that are laid. I'll walk you through all of these structures and their importance historically, but also what it means for how we understand protein folding and misfolding today. So on my right is the structure of lysozyme, which was one of the first structures to be solved of an enzyme and is a legacy structure from the Royal Institution. And as you can see here, it has been very nicely folded. So the amino acids from the primary structure, they interact with each other in various ways, and then they fold up to form the secondary structure and a tertiary structure. And this folded state, this folded enzyme is related to the function. So the enzymatic function, if this folding would not have happened, this protein would not be able to perform its function. Similarly, there are other proteins that also fold. One of the most abundant proteins is collagen, and here you can see it's folding from its primary sequence, which is the amino acid sequence, into a secondary sequence based on the backbone interactions, which lead to formation of structures like alpha helices and beta sheets. And then they come together and form a very complicated structure, which is the tertiary and then the quaternary structure. Now, how did this protein folding begin, and where does it begin? So it starts from the very beginning, which is your blueprint, which is the DNA, which has the code for the amino acids. So it transcribes that into the mRNA, and then the mRNA translates it from the ribosome into the protein. And that protein comes out and folds. So as you can see here, this is something that it looks like. It comes out from the mRNA, and then it would fold. And energetically, proteins have to be very stable in order to function. So the energetic barriers, they basically have they go down to the entropy, form these bonds, and then the native structure of the protein, which is basically the folded structure, is very, very stable. Now, sometimes proteins can misfold as well, which means that they may undergo some conformational changes while coming out of the ribosome and folding, or in the cellular environment of the cell. And this misfolding can lead to proteins forming clumps or aggregates. And these aggregates are what are observed in the Alzheimer's patients' brains. So these aggregates basically can be formed by any protein, and that can lead to either toxic gain of function or the loss of its original biological function. Now you can imagine that if this structure of this lysozyme were to aggregate, then originally it performs its function, an enzymatic function, and if it were to aggregate and form clumps, then all of this area that it has access to perform its function, it will not be able to do. And that would lead to its collapse of function and loss of its biological function. So going back to the ribosome, where all the the polypeptide chain is coming out and co-folding, and then eventually folds in the environment of the cell. So there are that means that there are different stages of the protein, just when it comes out from the ribosome. So you have basically the native state, which is the state that performs the function. But then there are also these transitional states when it is folding that are oligomeric in structure, like basically a number of single monomers of proteins coming together and forming these oligomers, or they would misfold and form more and more aggregates. Now, there are some other proteins that are present inside the cell that help proteins fold, and they are the chaperones. And in case misfolding is happening, you might have these chaperones that come together and cause the proteins to fold properly. Now, all proteins can aggregate. So it is not just the proteins that are involved in diseases like Alzheimer's that are aggregating, but essentially even this lysozyme can aggregate. So many years ago, in the 70s, Professor Chris Dobson had shown that if you were, one of his PhD students, if you were to leave this protein in a test tube in the lab and come back after many, many weeks, what you would observe is that this protein has formed gel-like structures, which means that under certain conditions, this has precipitated out of the cell. Now, what that looks like from a very physical point of view is that if this were your cell, and the blue color that you see here is the protein, in a normal state, the protein would be soluble. So you would not be able to see any crystals or anything that is coming from it. And at this stage is when your proteins are performing the right function, they're folded, and they're doing what they're meant to do. However, under certain conditions, things can change. So again, this is your cell, and we know that 70% is water. So we're going to try to see if other conditions can cause our proteins to, like what really can happen. So if these were the proteins that have come out from the ribosome in a different quantity, in a different concentration, meaning that due to some mutation, which could be genetic or environmental based, the code that transcribed from the DNA to the RNA has gone chaotic, has led to either an overproduction of your protein. Then this is what's happening, what's going to happen. You have an excess amount either of the protein, or the protein is not folding properly because it has some error in it, in its code, and it's going to just precipitate out or form these kind of goopy structures. And this is actually what's happening in disease. Even this is something that's happening during aging. And aging, viral diseases, that the protein homeostatic machinery is all gone wrong. And eventually, when the cell cannot take up the burden of all of these things going wrong, then what happens is basically everything precipitates out. Or most of the things and these could be different proteins. So a way to think about it is also like if you're mixing sugar, like we're mixing basically sugar in water, and after a certain concentrations, they're all precipitating out. So this is exactly that curve where you see that it reaches a critical concentration, and after that, it precipitates out. And what we see here are the formation of monomers, oligomers, and fibrils. And this is what is happening in case of Alzheimer's disease as well, where you have all of these different species that are formed. So this structure that we talked about, lysozyme, can also follow just the same trajectory and form monomers, oligomers, and fibrils, and lose its original function of providing, as an enzyme, fighting various bacteria, viruses, and fungi. So when do proteins misfold? In the laboratory, by changes in pressure, temperature, pH, salt solutions, that can cause these proteins to come together and misfold, or in disease, in case of cells and tissues, where exactly this mechanism is happening because of either genetic changes or environmental changes. And because it doesn't happen to all of us on a day-to-day basis, definitely nature, over the course of evolution, has taken care of this by providing certain protein quality control mechanisms. So in a healthy state, these protein control mechanisms balance it out, and the proteins do not misfold and stay in this soluble state. However, during disease, due to mutations or environmental changes, which we need to understand more, this balance is tipped off, causing disease, protein misfolding diseases like Alzheimer's, Parkinson's, ALS. What do these protein quality mechanisms look like? So this is a very complicated slide that you see here, but in essence, what really is happening here is there is a protein, a functional protein that is soluble, that is sitting right there. If it were to aggregate, the chaperone system would protect it, meaning that it's just gonna help and be like an escort to the protein and make sure that it is folded properly and performs its function. In case the protein misfolded, then the cell has a system which is just like a trash system where the protein is handed over to it, and then the misfolded protein is cleared out. Or it can also be phagocytosed and then eventually destroyed. So the cell makes sure that proteins that are aggregated, they are either folded properly or just thrown away from the cell. And definitely, this is not what is happening in case of Alzheimer's, because we are having so many of these aggregated proteins building over and over. And just the case of, you can imagine that if you were to leave your garbage in a room over for many, many months, it's going to take more energy and more effort to clear that out. And you can think that in terms of developing drugs as well, that if you have that garbage accumulated over many, many, many years, and the disease has progressed where the load is so much, it's hard to remove that by just one molecule, or it's hard to target that by just using one strategy. And that is something that we we want to understand how we can do it. If we could activate all of these protein quality control mechanisms by some way, then it would look like a fountain of youth where, as we have this accumulation of proteins, if we could just jump into a pool which had these rejuvenating factors that could solubilize these proteins or reverse their path from misfolded to the folded state, keep them in the soluble state, then we could come out from the other side with a perfectly soluble proteome. And this aggregation does not just begin anywhere. So if you were to look at all the protein misfolding diseases like Alzheimer's, Parkinson's, ALS, aggregation begins in one part of the brain. So I'll focus here on Alzheimer's. So aggregation starts off in the entorhinal cortex, so just somewhere deep here, and then from there, which is responsible for your memories, and from there to the hippocampus, and then it spreads out over time, which you can see on the x-axis here, which you can consider as age, to other parts of the brain. And that leads to, taking away of the cognitive function, cognitive decline, eventually motor activities, and eventually leading to death. Now it starts off, all of these are protein aggregates, but it starts off in different parts of the brain. So for Alzheimer's, it's entorhinal cortex. For Parkinson's, this aggregation happens in the dopaminergic neurons that are responsible for motor activities. And that is why you would see that in Parkinson's patients, it is not memory in the first case instance, but it is actually motor neurons that get affected. And that is what is very, very interesting, because that points out that there is something in the microenvironment of the cell that could be pushing it towards aggregation, which means that here you have your cell where all the proteins are nicely aggregated, but maybe it's not this blue protein that is causing the aggregation. It could be some other factor that just led to this goopiness, that just pushed the equilibrium to a side that caused things to aggregate. So in my work, back when I was doing my PhD, I was assigned this task of reversing this protein aggregation, which is taking it essentially from this state to this state. And I thought the best way to do it is to design small drug-like molecules, and because everyone likes to take small pills, drugs, and something which is easy enough, one pill cures all. So I thought, let's do that. So I designed a small molecule library that can bind to these various aggregates and slow down its aggregation. That is, leading from this state to this state over time. And but what I observed was that, A, the trash is too much. Even if I design this, if I have this one molecule that works in the test tube, I'm not sure if it's really going to work in human patients, because these plaques have already built up over a certain level. And in that case, there must be something else, because it has been seen that aggregation begins about 20 years before presenting itself in the clinic, which means the same curve that I showed you, which is the critical accumulation of these aggregates, begins much early on during aging. And there must be something that is happening that is pushing these proteins to aggregate. And then I looked back into my drug molecule library, and what I saw was that these drug molecules had similarities to small molecule metabolites. And that was very interesting, because I wondered that if, as synthetic chemists, we can design small molecules as drugs, then nature over its course must have already done so by having these endogenous metabolites. They're also small molecules like glucose. Here we have palmitic acid, which is again a heritage model from the Royal Institution. And this is vitamin B12, which is also a metabolite. And this comes from the structure from Dorothy Hodgkin. And these all metabolites also influence proteins. But we don't really know how they do that. And we know that metabolites change by our interaction with the environment. So overall, it's called as the metabolome, which changes over time. And this metabolome, these metabolites that perform various signaling actions or changing conformations of proteins, they keep interacting at various stages with the genome, which is basically the blueprint, and then having some regulatory functions, and also the proteins, which can change its conformation. And that is what we were interested in. And the question that we asked there was, just like how small molecules can change the aggregation of proteins, can our metabolites also change the aggregation of proteins? Another inspiration that came for this study is, and for study to see if metabolites are indeed doing this, is from fishes. So George and Somero, it's a beautiful book called Biochemical Adaptation, if any of you all are interested in reading. And it talks about how the environment influences proteins. So in fishes that are across different geographical habitats, it's seen that they maintain their life, they maintain their homeostatic processes, whether they are deep under, like in the ocean, near the ocean bed or up or near the surface, not by changing their proteins or the protein structure or how the proteins interact with other proteins, but by modulating the levels of metabolites, such as TMAO and glycerol in this case. And that is a very interesting observation because these fishes have different lifespan and they adapt to their environment in different situations and different habitats. And one way to think about this is also for humans because remember, we're not thinking about one size fits all. We're not thinking about one drug that fits all. We are very diverse, and we come from different geographies, having slight genetic changes in our genomes as well. So, and there are only 30,000 proteins, and proteins need to if they're not in their right structure, they're not going to function. So that tight coordination either will happen either by changes by either mutations, which is like intrinsic changes, or by changes that are leading through external factors. And it has been seen that these changes do lead to either health or disease. So these could be exercise, either temperature, food, diet. Diet has an influence, different microbiota in the gut, and aging, and other stochastic factors that we do not know. Another way to think about this is people living in the blue zones of the world, where people live much longer and do not have age-related neurodegenerative diseases. The only factors that, apart from their genetics, that do play a role in that is their lifestyle, which comprises of diet, exercise, community, and how they're interacting with each other. And it's still like we're doing many doing research to find out what these mechanisms are, how that contributes to extension. But definitely it is bringing about a change inside the cell. And here we postulate that it is not just the proteins, but also these small molecule metabolites, because they're all existing inside the cell and basically affecting each other's homeostasis that are causing these changes. So that's what the cell looks like. If you were to take the prokaryote E. coli and look at its proteins inside, it's in a crowded environment. They're so close to each other. So they can basically they're basically just sitting at the edge to move over this concentration. So one of the papers that was published a few years ago showed that proteins that aggregate in these diseases like Alzheimer's, Parkinson's, they're basically sitting at the edge, ready to aggregate. And if there's any perturbation that is happening, it's just going to flip it over and cause them to aggregate. Now, the crowded environment of the cell does not have just proteins. It also has metabolites, lipids, a whole lot of ions, many, many things. And that's also like sitting at the edge, just ready to, if something goes wrong, just ready to aggregate. So what we did was we asked that, can we map out all the processes that are dysregulated in neurodegeneration? So we looked at many, many, many studies across the literature to map which metabolites or which biochemical processes are dysregulated in neurodegeneration, and in particular, Alzheimer's. So most of the literature that's out there has been dominated by the amyloid hypotheses, the aggregation hypotheses, and lots of papers show that we have tau aggregates, we have amyloid aggregates, but there are also a lot of biochemical pathways that are dysregulated. So what we saw was that all of these pathways had these distinct metabolites that are present that are also dysregulated. Meaning that in patients having these disease, if their cerebrospinal fluid was taken, then it was found that when compared to a normal patient, they had an abnormal level of these metabolites. So we thought, why not look at these dysregulated metabolites and see whether these metabolites have any effect on aggregates or the process of aggregation. We use the model worm because, of course, we don't want to kill humans, and we need very, very large numbers to get something which is significant. Basically, N, which is a sample size of thousands, rather than just 5 or 6, because again, we may not get something that is significant. So the worm, which is C. elegans, it's a nematode. It lives in the wild. We can culture it in the lab. It has a very short lifespan of 2 to 4 weeks. It's a powerful model for studying the biology of aging. It has about 40% overlap with human aging pathways. And it also, you can express human proteins in it and see how what really happens during aging, how they aggregate, and whether we can have some interventions or not. So we took our dysregulated metabolites, and which were basically found in these dysregulated in neurodegeneration, and what we saw was that these dysregulated metabolites, when supplemented, they can activate the protein quality control mechanism of the chaperone response, the heat shock response, that can prevent this aggregation from happening. So this protein that sits right here across the table in front of you, which is amyloid beta, here you can see it's a very flat structure. This protein in the cell does not fold like lysozyme. So it doesn't have one structure. It has many, many, many thousands of structures that can be. So if we can, and many molecules of this can come together and aggregate. So what we showed was that a protein quality control mechanism of the chaperone response can prevent the aggregation of this protein. In another study, taking together different metabolites here, which is vitamin A and vitamin E. So we have a structure here of vitamin A. You can see the size of the, oops, I dropped an atom, sorry about that. So that could happen in the cell too, not the atom, but maybe the molecule. So yeah, errors. So here is vitamin A, and what we saw was that if you have this vitamin A, and just imagine vitamin E, in certain concentrations, it can have a zero-sum effect on protein aggregation. Now, what does that mean? So A-beta can aggregate faster under some conditions, and it can aggregate slower under certain conditions. So this vitamin A inhibits its aggregation if I just supplement vitamin A. But if in the same mixture I also add vitamin E, then this aggregation goes back to 0, which means that I have two metabolites that have opposing effects, and at the right concentration, they just keep the protein soluble. So you can think of it in terms of this same pot, the same beaker, where everything is soluble, and to maintain that solubility, you just need the solutes to be in the right concentration. And in this case, the protein, amyloid beta, and the vitamin A and E in the right concentration. And the interesting part is that we haven't studied all of these molecules, all of these vitamins, starting from historically Dorothy Hodgkin's like vitamin B12 structure. And structure information is super important because that allows us to understand how these molecules really at the atomic level interact with the big protein molecules. So systematically, we made this platform where we can understand how these metabolites keep the protein stable and metabolites can keep these proteins stable. We use sophisticated other methods, such as mass spec-based proteomics, which basically allows you to study quantitatively the proteins inside a lysate, like in a big complex, where we can find out if the structures of the proteins change or not in a very complicated environment or not. And what we found was that the metabolites that are dysregulated, they indeed change conformations of the proteins that eventually aggregate in Alzheimer's disease, which is basically the amyloid beta. Not only amyloid beta, but what we found was that the metabolites that are dysregulated in general in neurodegeneration, they also cause conformational changes in other molecules, like alpha-synuclein, and also some of the protein quality control mechanisms, which gave us an idea that, that metabolites have an influence on protein quality control mechanisms and also like protein structure, and that could be something over a period of time that is causing these proteins to aggregate. So it's almost like this buildup is happening again and again and again until the cell says, okay, not enough, and then the aggregates just start building up. So we showed that endogenous metabolites can modulate protein aggregation, but what does that really mean in the context of protein misfolding diseases? And that is something, again, which is very interesting and part of our current work. If we were to isolate, like if Dr. Alois Alzheimer now decided to go back to isolate these plaques from different patients' brains, what he would find is that all these plaques are not in the same, they do not have the same structure. They are different. So the plaques that come from familial disease, which is genetically inherited, are different from the ones that people just develop sporadically. Also for Alzheimer's, another key fact is that it's about more than 90% of it is sporadic. That means it doesn't have a genetic, like a heritable component to it. And the structures that come from both are different. So that again points out that there is something that is pushing these structures to form and be different. So many labs across the world have shown that when you isolate these structures directly from the brain, they are different. And we thought that maybe it is the metabolites that are pushing these structures to form and having different conformations. So the disease variants are different. Again, you must have also heard about people like finding that, especially the very famous Nun study where they had found that there was a lot of amyloid plaque in the brain, but it did not cause any, you would not see the correlation between the amount of amyloid in the brain to the cognitive decline. And that is because all the plaques do not cause the disease. It's almost like thinking about it in terms of the malignant and benign cancer. And here you have the off-pathway and on-pathway aggregates, which again, your protein starts from its soluble state, the monomer, and goes on to the on-pathway, leading to more toxic aggregates, which are different from the off-pathway, which are non-pathogenic aggregates. So that also means that we cannot just have one drug that really that is going to clear out all of this trash, because the aggregates have different structures. And what changes during a person's lifetime is metabolism and aging due to aging. And that is how we combined our observations from the metabolites, which we studied that modulate protein aggregation, to actually disruption and changes in metabolism over aging, leading to the onset and progression of protein aggregation. And the small molecule metabolites being the key players. And that brings me to highlight that aging is one of the highest risk factors for neurodegenerative diseases like Alzheimer's, Parkinson's, ALS. And what we think of aging as just like this glass of beaker of water where you have proteins that are soluble and overage at some point because of the loss of the protein quality control mechanisms precipitating out of the solution. And this is indeed seen back in our favorite model organism, C. elegans, where if you were to take the whole proteome, the whole proteins, all the proteins from a younger animal, which is day 2, and you would take all the proteins from an older animal at day 11 or 12, you would find that there is more aggregates, more insolubilome that is found at day 12 of the worm. And this showed that, this shows that aging contributes to loss of your protein homeostasis and protein quality control. So what can we really do about this? What can we do to increase our health span in the context of aggregation, or when we know that these quality control mechanisms are going down? One of the things that has been seen that brings about a change in metabolism is caloric restriction. So in worms, many, many thousands of worms, if we give them less food. So in the lab, they love eating this microorganism called E. coli. And if we just don't give them any of this bacteria, basically they just end up living twice as long. And that's amazing. Like, I'm not suggesting any of you all to starve yourself. Please don't do that. Food is important, and it's one of the things that we all enjoy a lot. So, but what I really mean by this is that your dietary intake has an impact on the molecular processes of our body. And in the worm, we have seen that basically processes like ribosomal biogenesis, which is the same ribosomes, if you remember from one of my previous slides, that are causing these proteins to fold. That ribosomal biogenesis is being affected by calorie restriction, meaning that if you're changing the diet, the ribosomes are formed in different amounts, and that is contributing to protein folding or misfolding. And that gives us an open area to investigate that what are the mechanisms and what are the metabolites that are there in caloric restriction that are causing changes in lifespan and health span. Now, caloric restriction is not just, these studies have not just been done in worms, but it has been seen across species that they have beneficial effects on increase in lifespan and health span. Another thing that is really helpful is exercise and movement. Any kind of movement brings about a change in the mitochondrial biogenesis. And again, even in the mitochondria, you have a whole range of metabolites. You have adenosine triphosphate, the energy currency as what we call, and then you have NAD/NADH ratio, which changes the pH of the cell. And exercise and movement can bring about changes in these metabolites. Overall, big studies like longitudinal studies have suggested that exercise, movement, dance can keep people healthier, much healthier till the end stages of life. So what we really need before we find therapies is diet and movement-based interventions, because we know that metabolism is changing over aging. And metabolite-inspired therapies for age-related diseases, which really means is that we're not targeting the end-stage aggregates. We're not really targeting the end-stage amyloid that's being formed because it's too late, there's too much trash, but we are targeting something much early on. So if we can find the mechanisms of calorie restriction, we can find out the mechanisms, we can find out how those metabolites are really interacting with the protein quality control machinery, we can come up with therapies that are metabolite-inspired and that rejuvenate the cell back into a more younger state so that the aggregation does not happen. There are a lot of longevity clinics that we see have been spinning out like in many, many countries. And they can detect early biomarkers. And these early biomarkers are also in the form of metabolites, which can track down how are these changing over the age. And what can people do to change these biomarkers over age and basically can act as a preventative measure for for age-related neurodegenerative diseases. And it is not just confined to Alzheimer's, but also other diseases like cancer, which happen to, which has a correlation to age as well, and also COVID, because as you must have seen over the last couple of years, when people were being age was being one of the factors that was heavily seen, that people at a higher age were more affected by it, and also there have been we don't know what the long-term effects are going to be, and it could be that they have different proteinopathies that are coming up later on. So taking all of that together, the UN also recommended back in 2020 as the decade of healthy aging, and that is what we want to emphasize here, which is that it is like we are looking at the molecular mechanisms here of diseases, but what really happened, what really, the basic basis of understanding these is to promote healthy aging. And the fact that we know that diet-based and exercise-based interventions can do something and help us age in a healthy way, that is something that we can do now before all of us are coming up with different drugs to target these diseases. So with that, I'd like to thank the different groups that I worked with. As such, science doesn't happen alone in the lab. It needs a big force and many, many, many scientists. As you can see here, it is across 3 countries and 2 continents where some of the work that I performed was done. And basically, it's also very interdisciplinary. So it's not just biology. It's not just like the disease is the observation. We have used different tools that are coming from physics, chemistry, computation, mathematics, and we're combining all of that to find out these key rules, key principles underlying these diseases. And thanks to the government for funding these research. So that all goes for our taxpayers' money. The money that we go in goes to the research that we're performing. And with that, also we have this metabolite homeostasis consortium, because this field is, although we know that metabolism changes and metabolites exist, there are not many systematic studies towards it. So in this consortium, we are bringing together scientists who are working on not only the disease aspect of metabolites, but also how metabolites can come together and form materials just like proteins do. And try to find out more and more information about it. And I'd like to dedicate this lecture to my father, who passed away 12 days ago. And he suffered from age-related lymphoma that happens in males over a certain age. And one of the things that we used to discuss was healthy aging in the context of my research. But sadly, he went away too soon. He loved cricket, though. So with that, I'd like to actually do a little game here or exercise here, which is to leave you all with something that you can go take forward beyond the science that we have discussed here. Does anyone dance here? I'm sure all of us do in the confines of our homes, but if there are any salsa dancers or Latin American dancers here. Okay, so we have one. Is there anyone else who would like to join? Okay, I'll ask Hassan to come here because I know that he... Can we have you as well? So, yeah.
You teach me?
No, can we just teach everybody a few dance steps so that they can go back with it?
Well, I didn't sign up for this.
Well, we have a discussion round, so we can discuss all the science. But now that we know some aspects that, of the science that lead to these things that we can actually do in our day-to-day life, then we should do it. So why not? Can I please have you here? So when you go back home today, over the week, we'll have the two of them. Do you do salsa?
A little bit, yes.
You do salsa?
2 lessons.
Okay, amazing. Can we teach everyone just 2 basic steps?
Of salsa?
Yeah.
Do you know how to do salsa?
A little bit, come on, go on.
Go on.
So
Do you want to take the lead or? 2 lessons.
Why don't we tell them first the basics front? Well,
Okay.
this side, so, is there any music by any chance? Anyway, so salsa basically is a step to the side, and then, I'll follow you, there you go.
I'm doing great, I'm doing great. Can you get some music on?
Ooh, thank you. Thank you so much, Asa. So I hope you all remember these dance steps so that you can keep following that over the next few days, and that encourages you to go back to dancing or go start dancing. And I'd just like to share another anecdote or another story which could inspire you from this dancing. So back when I was in Cambridge, I used to do flamenco, and I did not find a place to dance. And my friend took me to an old age home where we thought it had nice wooden flooring, and we started dancing. But the place, all the people who lived in there were totally annoyed, and they did not like the sound. So they decided to gather around us and scare us away, like just push us away. But then when they came and they saw us dancing, they all got intrigued. And the doctor in the care home told us that, why don't you just perform something for them? So then we started dancing, and flamenco has music has a rhythm, and your brain can catch the rhythm. So if any of you all have studied neuroscience or read about it, it's like your brain can rewire with these kinds of movements. So we were doing this dance and clapping, and there was this woman sitting in front of me. I was told that she was in her late 80s. Her name was Judy. And she had traveled around the world back in her day, studied, and now she had late-stage Alzheimer's, and she couldn't move. So this almost sounds too good to be untrue, too good to be true, but this is what happened, was that when we were clapping and dancing, so me and my friend, she actually just livened up, and she did this from her wheelchair to us. And that was like one of the moments that made us think that, our research that we are doing, it's very interesting, very important, but it is taking its own time to reach the public. So these are little things that do have an impact. And this is research that, when you go back, I'm sure you can do a very systematic search on Google Scholar and not find any misinformation there. But these are things that do lead to healthy aging. So this was like a true story that kind of inspired that if we cannot, like while we're working on our research, there are some things that we can do over a lifetime, over the immediate lifetime, that can actually help us to age in a better way. And that is what, as communities, as countries, we need to focus on. And I think that is what the UN also means by the decade of healthy aging, where together as citizen scientists or as communities, we can make these changes in our, we can all work together as scientists, to bring this out together. And with that, I'd like to open up for questions, debate, critique, or any things that you think that I don't believe in this, we can talk about it.